The last set of plant notes will be on reproduction. At some point in evolutionary history, there were modifications in the way plants were able to reproduce that allowed for them to be adaptable, to be able to spread to a variety of land habitats. These modifications were key in the success of plants. Originally, water was used to spread the gametes. Remember, a gamete is a sex cell, like a sperm or an egg, for example. But this was replaced by other methods of distributing these gametes, such as wind and animals. The embryos became protected in seeds, and so that allowed for the seed to have a wide variety of protection to survive harsh environments. This is something that we talked about in a previous set of plant notes, how important this actually is for angiosperms. There are some plants that can reproduce asexually, but we will focus on sexual reproduction. Some plants can actually do both sexual and asexual reproduction, but again, our focus in plants is going to be on their sexual reproduction. After a seed has germinated, a young plant is going to start to form all of its major organs. So we're going to have roots, we're going to have stems, we're going to have leaves as it develops. And these are considered to be vegetative structures. A vegetative structure is a structure that is not used for reproduction. So again, since roots, stems, and leaves are not used for reproductive purposes, for sexual reproduction, these would be considered vegetative structures. So you're probably very familiar with the term vegetable. But as a botanist, they would have a very different definition of a vegetable versus a fruit, which is a reproductive structure for a plant, than a typical person who goes to the grocery store. Right? So when you go to the grocery store, you might think that you're buying vegetables. For example, zucchini. You might consider zucchini a vegetable because it's not sweet. And usually fruits are associated with things that are sweet. Or you might say a tomato or an avocado are a vegetable, again, because they're not sweet. However, as a botanist, they would say that those are fruits because they have seeds, right? So they are used for reproductive purposes. If it's used for reproduction, a botanist would not consider that part of the plant a vegetable or a vegetative structure. They would consider that a reproductive structure or a fruit. So for example, in lettuce or spinach, they would consider those leaves as vegetables. At some point, a plant does need to be able to reproduce. And so in order for an angiosperm to reproduce, they're going to use flowers. And so that means that the vegetative structures have to somehow transform into reproductive structures, into reproductive flowers. And this is going to happen when the apical meristems and the shoots start making flowers instead of making leaves. This is going to be due to changes in gene expression, gene regulation. Some genes will be turned on, some genes will be turned off, and this will cause the apical meristems to go through changes in gene expression. They will no longer make leaves in that moment in time. Instead, they will start making reproductive structures like flowers. You don't need to know the mechanism of how, you just need to know that it does involve a change in gene expression at the apical meristems. When this occurs, there will be 4 sets of what are considered to be modified leaves. Remember, we used to be making leaves from these areas, but now we're making flowers. And so these would be considered modified leaves. There can be sepals. And well, first of all, we're going to focus on a flower that looks like a pretty typical flower, but please understand that not all flowers look exactly like this. This flower is said to be hermaphroditic. A hermaphroditic flower contains both male and female reproductive parts, which means it is capable of sexual reproduction within the same flower, so with itself, which we'll get into more details coming up. But not all flowers look like this, right? There's a variety of flowers out there, very diverse amount of flowers out there. So we are looking at like a typical flower. So starting with sepals, on this flower, the sepals are down here. And what the sepals do in regards to their function is they protect the bud. So before the flower actually blooms, it will be protected inside of a bud while the parts of the flower continue to develop inside. And the sepals are going to be those, they look like leaves kind of, when the flower is actually still closed. The petals are brightly colored. They're pretty obvious. And they're often very scented. They might smell very sweet. And these are going to be used to attract the pollinators. That's their functions. The stamen, those are the male parts of the flower. In this picture, it would be like one of these entire structures. There are different parts to the male part of the flower, which we'll focus on an upcoming slide. But this is where the male gametes are going to be produced, is within the male part of the flower, the stamen. And so the male gametes ultimately will be the sperm. For carpels, those are the female parts of the flowers, and so it's going to be this entire structure right here is considered the carpel. It also has several parts, which we'll go over on upcoming slides, but it does contain what's called the ovules, which are ultimately where the female gametes or the eggs will form. So in this case, this one has one ovule right here, but some flowers have more than one ovule. Here's another picture of a flower. And so all the parts are shown on here as well. One thing that students often get confused on is ovule versus ovary. The ovary is going to be this bottom part, this bigger part right here of the carpel. So it's part of the female structure and it contains the ovules. Inside of the ovary, there will be ovules. There's usually many ovules. This one has 6. Each ovule can potentially form a seed. Okay, so in this picture, we have one ovary, and inside of that one ovary, 6 ovules, and so 6 potential seeds could form if they became fertilized. Here's another picture, same thing, right? So we have one ovary, 6 ovules inside, and those 6 ovules could potentially form 6 seeds if they were all fertilized. Another structure that's important to flowers that is not a modified leaf are going to be glands that they have that are typically found deep within the flower itself, like towards the base of the flower down here, and these are called nectaries. These nectary glands make a sugary solution called nectar, and that's used to feed organisms that depend on flowers for their food. It says insects right here because insects are a main pollinator, but there are other animals that could be drinking the nectar as well, like a hummingbird or a bat. Bat has a really long tongue that can get in there and drink that sweet nectar. The male part of the flower, again, is called the stamen, and it is composed of 2 parts. The stamen on the top right here has what's called an anther. The anther is where meiosis actually is going to happen for the male part of the flower. Remember, meiosis is the type of cell division which starts with diploid cells and makes haploid cells. These haploid cells are considered gametes, so they're only going to have half the genetic information. Ultimately, inside the anthers, eventually, that's where the sperm will be made. The filament is going to be this long structure that holds that anther up. So its job is to provide support for the anther itself. Let's quickly review meiosis because it's probably been a while since you've been exposed to this level of detail. We did learn meiosis in Bio H. Remember, meiosis is a special type of cell division. It's different from mitosis because meiosis only occurs in the reproductive tissues of a sexually reproducing organism, and it results in gametes, okay, 4 haploid cells that are called gametes. In the process of meiosis, we'll start with a diploid cell. It will go through 1 round of cell division to make 2 haploid cells right here. Those two haploid cells each go through another round of division called meiosis II, and ultimately we will end up with 4 haploid cells. There are some key events that do occur in meiosis. One of those events is happening in prophase 1. In prophase 1, the homologous pairs of chromosomes are going to match up with each other. They're going to form what's called a tetrad. So here's a tetrad, here's another tetrad. IB also likes to call that a bivalent, bivalent, a bivalent. And what that is it's a homologous pair of chromosomes that are coming together. So if the red ones are the maternal chromosomes and the blue ones are the paternal chromosomes, then they represent the matching set of chromosomes. So for example, chromosome set number 1, and then here would be chromosome set number 2. And so they line up as homologous pairs, they form the tetrads or bivalents, and then you can see there are parts that are touching on each homologous pair set. That's where they're going to swap genetic information. Okay, so that's called crossing over. They will swap genetic information. This means that a little bit of the maternal chromosome goes onto onto the paternal chromosome, and a little bit of the paternal chromosome goes along the maternal chromosome. So it kind of shuffles up the genes, and this is okay because they are homologous genes. So for example, if mom has the big A and dad has the little a, then the big A can swap places with the little a. But because A still codes for the same gene, then that's fine. And this shuffles up genetic diversity. It's one of the things that ultimately leads to unique daughter cells at the end of this process. So that's one of the major things that happens, crossing over in prophase 1. In honors, we did talk about independent assortment in metaphase 1. So homologous pairs line up randomly along the metaphase plate. They will then separate as pairs in anaphase 1. So pairs, half the pairs move to this side, half the pairs move to this side. So at the end of meiosis 1, we will have 2 haploid daughter cells. These daughter cells are haploid. They're not diploid daughter cells anymore. Don't get fooled by the X. You need to start with counting how many chromosomes you started with. We started with 4, 1, 2, 3, 4. That's a diploid number of 4. We are going to end with haploid cells because they only have half the genetic information. This cell only has 2. This cell also only has 2. And so these are haploid cells. The reduction in chromosome number happens in meiosis 1. Meiosis 2 is considered an equal division because we're going to start with haploid cells right here in prophase 2. It's going to go through single line of chromosomes aligning along the metaphase plate, not lining in pairs, right? So you can see pairs here, no pairs here. Sister chromatids get pulled apart in anaphase 1 into individual chromosomes. And so at the end of the process, we'll have 4 cells that are still haploid. Count the chromosomes. 1, 2, 1, 2, 1, 2, 1, 2. So 2 is half of what we initially started with here, which was 4. So therefore, these are still haploid. The chromosome number does not get halved twice. That's because the chromosomes duplicated in interphase. That's why we had an X. So being able to follow through this process is definitely important. And they're all genetically different from each other, right? Due to crossing over, due to independent assortment. Okay, what eventually forms in male parts in the anther is a pollen grain. And this pollen grain is going to be made up of 2 haploid cells that were made inside that anther. So the anther makes the haploid male cells in meiosis, but then it forms a pollen grain from those haploid cells. So some of those haploid cells are going to be called tube cells, and some of those haploid cells are going to be called generative cells. The generative cell is going to move inside of the tube cell, and this is a pollen grain. So you can see here we have our tube cell shown in blue, and then here's the generative cell. It moved inside of the other haploid cell. Collectively, this is a pollen grain. Eventually, this generative cell will divide by mitosis to produce 2 sperm, 2 more haploid cells. So it does not stay as 1 haploid cell inside the tube cell. It will divide to make 2 haploid cells, the sperm. So we're going to have 2 sperm. Oh, sorry, going back. This is important because angiosperms are going to go through a process known as double fertilization. So they're going to need 2 sperm in order to do that. I will go over that in more detail in a bit. Pollen grains are incredibly tough. They have a tough outer coating that helps them to stay strong as they have to be bounced around throughout a harsh environment until they hopefully make their way to the female part of the flower for pollination. And so they are protected by this tough coat. And each coat is different in each species. And so that's why you could be allergic to some pollen and not allergic to other pollen potentially, because what you're allergic to is that outer coat of the pollen grain itself. This shows a cross section of the anther. So that, remember, is where meiosis occurs. So meiosis is going to occur in 2 spots in angiosperms, and one of those spots is going to be within the anther itself. So inside of the anther, it will go through meiosis 1, meiosis 2, and ultimately we will get our pollen grain, which is going to have a tube cell with generative cells inside, and those generative cells that are inside will ultimately divide to make the 2 sperm that will still be inside of our tube cell. The female part of the flower is called the carpel. IB calls it a pistil, but AP calls it a pistil. You have to call it a carpel. Carpel is the IB term. There's very few differences in AP and IB terminology, and this is one of them. So you must call it a carpel, not a pistil, even though they are the same thing. The carpel has 3 parts, the stigma, the style, and the ovary. The stigma is on top, so right here, and it's usually considered to be kind of sticky because its job is to trap the pollen. So I usually say the sticky stigma. The style is long, and it supports the stigma. It holds that stigma up high, as well as connecting the stigma to the ovary itself. So maybe think of like something long that has style, like a high heel, right? Has style. They're said to be stylish, and so that long heel of a high heel, maybe that will help you remember. The ovary is down here, and this is where the ovules will be inside of it. So you can see this one has 2 ovules, 1, 2, inside of the ovary. And this is also where meiosis will occur for the female part of the flower. So remember, meiosis occurs in 2 sections of a flower. It's going to occur in the anther to make the male gametes, and it's going to occur in the ovule to make the female gametes. Here shows you a different picture of an ovary, just so you can kind of see that they're not always like big and round and bulbous like they're usually shown in diagrams. They can be slender. They come in all different shapes and sizes, just like flowers do. Okay, so quick review. Here's the ovary, and then there is an ovule. The ovule is what is going to undergo or contain the cells that undergo meiosis. So there's going to be a diploid cell in the ovule that can undergo meiosis. This diploid cell divides by meiosis 1 time and produces our 4 sex cells. There is something that's a little bit different in females versus males. When meiosis occurs in males, they actually make 4 completely usable gametes. So all 4 gametes are equally likely to continue to develop in the process. But in females, that's not true. At some point during development, all the cytoplasm and other important nutrients go into 1 of the 4 cells. And so only 1 of them becomes a usable cell. In this case, eventually forming an egg. The other 3 are completely useless. They're called polar bodies, and they do nothing. They're usually reabsorbed into the female reproductive parts. So again, females will only make 1 usable sex cell, and the other 3, although they form, they don't do anything. They're non-functional. 3 polar bodies are non-functional. So then we have our cells that are made by meiosis, and so we get the 1 for females. This 1 cell is ultimately going to go through 3 rounds of mitosis. So we have our haploid cell. It's going to divide by 1 round of mitosis to make 2 haploid cells. Those will divide again to make 4 haploid cells. Those 4 cells will again go through mitosis to make 8 haploid cells. So we will end up with 8 haploid cells due to the 3 rounds of mitosis in the female part of the flower. Of those 8 cells, which you can see all 8, 1, 2, 3, 4, 5, 6, 7, 8, we only care about 3 of them. So we care about this one down here. That one's the egg. And then we care about these 2 in the middle. Those are called the polar nuclei. So 2 polar nuclei and 1 egg cell. That's all we care about of these 8 haploid cells. Pollination is when pollen lands onto the sticky stigma of the carpel. And that's really it. If it lands on it, we have had pollination. Pollination is important because it will hopefully lead to fertilization, which we'll talk more about in a bit. There are different ways that pollination can occur due to the spreading of pollen. Wind may help to carry the pollen. So you can see that here, right? The wind kind of blows the pollen all around. Animals like insects or other types of animals as well, like bats or hummingbirds, they can land on the flowers to drink the nectar usually. And then as they're drinking the nectar, they pick up the pollen on their like fuzzy bodies or feathery bodies, and that will allow for it to carry that pollen to the next flower, and that will help to promote cross-pollination, which is going to be pollination that occurs between flowers on 2 different plants. Plants also have the ability to self-pollinate, which means pollen from that flower can be transferred into the female part of the same flower or on a flower that belongs to the same plant. Oops, sorry, going back. So here shows you just a few ways. We have wind, we have hummingbirds, they can get it on their feathers. Here's a butterfly that can get it on its feet or its wings, and then a bat also has fur, so the pollen kind of gets stuck on the hair, and it helps to transfer from flower to flower, and then of course a bee. This one shows the difference between self-pollination and cross-pollination. There are 2 methods of self-pollination. It could be pollen from one flower falls onto the stigma of a flower that's on the same plant itself. That is still self-pollination, as well as within the same flower, that is self-pollination also. To get cross-pollination, we would have to have pollen go from one flower, from one plant onto a flower of a different plant of the same species. So at some point we will dissect a flower and you'll get to see all the parts of the flower up close. Let's keep going. Okay, so we've had pollination where the pollen lands on the stigma, and now it's time for fertilization. And so remember, a quick review, we have our pollen grain, which is a tube cell with the generative cell inside. And that is going to, the generative cell ultimately will divide by mitosis to make the 2 sperm. So you're going to see the double fertilization happen in fertilization. And then we also have, quick review, our ovule, which within the ovule went through meiosis. And then the 1 cell we got for female gametes goes through 3 rounds of mitosis, making 8 haploid cells. So you can see all 8, but remember we only care about these 2 polar nuclei in the middle and then the egg right here shown in orange. So after pollination happens and the pollen grain lands on the sticky stigma, the tube cell will form a tube. Surprise! So our tube cell forms a tube that elongates down the style and makes its way towards the ovary. While that is happening, that's when our generative cell, which remember is inside of our tube cell, goes through mitosis to make our 2 sperm. So now here's our tube cell, right, making our way down there, and you can see here is the 2 sperm cell. We need the 2 sperm cells because ultimately one of them fertilizes the egg to form the zygote. So remember, sperm is N, it's haploid. The egg is N, it's also haploid. So when they come together and they unite for fertilization, we end up with 2N. So we're back to diploid. This is how the diploid number is restored for sexually reproducing organisms, is through fertilization. So now let's switch gears and talk about the second sperm, right? Because we have double fertilization. So the second sperm is going to actually fuse with the two polar nuclei in the middle. And so that is going to have 3N tissue form. So N from one polar nuclei, N from another polar nuclei, and N from the sperm. That is 3N. We call that a triploid tissue. This triploid tissue is important because it forms what's called endosperm. Endosperm is like the food storing tissue for the developing embryo. Because remember, when all of this is happening, ultimately there has to be food available for this organism to grow before it can photosynthesize. So it's going to store up food in the endosperm. Then the zygote continues to develop into an embryo, and then the integuments, which is the outer layer of the ovule itself, will harden, and this is going to form a seed. Okay, so we have integumentary layers as well. Your skin is a major part of the integumentary system because it's your outer protection. And so this is true also for plants. They have an outer layer surrounding the ovule, and it's going to harden to help form a seed. And now this embryo is protected. While seed formation is also taking place, the ovary, which remember is around the seed or seeds, there can be multiple seeds depending on how many ovules there are, the ovary itself will develop into a fruit. And so again, this fruit may contain one seed or more than one seed. This depends on how many ovules were initially within it. But you're probably very familiar with some types of fruits. For example, an apple. Right? If you cut an apple in half, you'd see the seeds inside. Well, this fleshy part of the apple is the ovary. So when you eat an apple, you're eating the ovary. It developed into fruit. But not all fruit is going to be fleshy like this. Some fruit are actually dry. So you can have dry, hard fruits. The ovary can form a dry, hard fruit. For example, in the case of a nut, we typically buy nuts at the store that have already been cracked, so they're pretty easy for us to eat, right? Like this is a walnut. You might buy walnuts at the store like this, or pecans, or almonds that have already been shelled. But you can sometimes find, and definitely you used to find way back in time, that things weren't quite this convenient, and you would buy nuts in shells, hard shells like this, and you'd have to have a nutcracker to crack it open to be able to eat that seed. And so this is the fruit of a walnut. It is hard. It is not fleshy. It is dry and hard. So that's just a different kind of fruit. There is a genetic advantage to cross-pollination. Self-pollination leads to what's called inbreeding, which is when organisms that are very closely related to each other reproduce. And so that leads to less genetic variation, and less vigor, which means like strength for the organism to survive. And so in order to limit this or prevent this, which would promote cross-pollination over self-pollination, many flowers have mechanisms to help them do this. They may have their reproductive parts mature at different times. So maybe the stamen is mature, but the carpel's not. So what that would mean is the carpel's not receptive yet for the pollen. It's not ready to receive the pollen, even though the pollen is mature within the same flower, which means that it can only go to another flower for reproduction. And vice versa, right? Maybe the female part is receptive, but the male part's not mature enough to fertilize that egg. It could be a structural arrangement. So we've been looking at pictures of flowers where the stamen are tall and so is the carpel. So they're all about the same size, but this is not always true. Sometimes there are really, really tall, for example, female parts, and then really, really short male parts. And so this would make it more challenging for the pollen to make that leap way up here and attach to the stigma within this flower. Can it happen? Sure. But does it limit it? Yes. It's definitely going to happen a lot less than if they were really tall and right next to it. They may have male or female flowers, meaning they're not hermaphroditic flowers. For example, here's zucchini flowers, right? You see there is a male part on this flower and no female part. And then there's a female part on this flower and no male parts. So on the same plant itself, they may have strictly male flowers and strictly female flowers. So the more plants, zucchini plants, you plant in your garden, for example, the more chance that there would be of cross-pollination. So you can actually see that only the female only the female flower develops into fruit. Here's the zucchini forming. This is just stem. The male parts cannot develop into fruit. They are simply the pollinators. There are other types of plants too that have male and female flowers on separate plants. Like the entire plant is strictly male and the entire plant is strictly female. So that also promotes cross-pollination. Some plants have a biochemical block that actually prevents the pollen grain from fully developing, and so it never makes it down to fertilize the egg. So we'll talk about this genetic component on the next slide. This is called self-incompatibility. So some plants have genes that make them incompatible with themselves. And so ultimately what would happen is within this gene itself, it's too closely related genetically, and so it kind of rejects the pollen within the plant itself. I'll show you a picture on the next slide. If it has any matching alleles at all, then that pollen grain would be rejected and it wouldn't be able to grow to form the tube cell that's needed for fertilization. Quick review from BioH: an allele is an alternative version of a gene. In BioH, we talked a lot about genes that only have two alleles, right? Big A or little a. You're going to get two, but you could get two big A's, you could get two little a's, or you could get big A and little a. And so that's pretty normal to only have two versions, two types of alleles for a gene. But there are plenty of genes out there that are more variable than that. They may have multiple alleles. And we talked about examples of this too in BioH. For example, the ABO blood grouping system. There are three alleles that exist in the population: the A allele, the B allele, and the O allele. You're still only going to get two, one from mom and one from dad. But because there's multiple alleles, more than just two in the population, there's more genotypes that are possible, and therefore more phenotypes that are possible within the population also. There are many genes, though, that are much more variable than that. There could be 10 versions of the allele, 20 versions, 100 versions, 200 versions of a particular gene. And so the more versions, the more likely that self-incompatibility would promote cross-pollination over self-pollination. In this example, there are 10 alleles, right? So you could get the genotype S1, S2, S3, S4, S5, S6, S7, S8. S stands for self-incompatibility, by the way, S9, S10. Or you could get any combination of it. So you could get S1, S10, S2, S3, S6, S9, and so on, right? So lots of variation in genotypes, which means lots of variation within that particular phenotype for self-incompatibility for that particular plant. And that's only looking at 10 alleles. Remember, there can be many more than that. So let's look at this in a picture. This shows that this genotype of the plant is S1, S2. That's the parent plant. So if the haploid gamete has S1 for its allele, for example, then S1 is too similar because this plant already has S1. So the tube cell will be stopped. Same for S2, right? This plant already has the S2 allele, so the tube cell will be stopped. In this one, S1 is stopped, but S3, the tube cell gets to elongate and therefore fertilize the ovule. Here we go, S4 and S5, they both get to elongate and we get fertilization because it's not like the parent plant. So only non-matching alleles are able to form the tube cell and therefore fertilize the zygote. Seed dispersal. Many types of plants have diverse mechanisms to be able to disperse their seeds. Seeds can be dispersed by wind. I'm sure you've seen like a dandelion. If you ever pulled a dandelion out of the ground and made a wish by blowing on it. So what you're really doing is blowing all those little dandelion seeds everywhere, and that just leads to more dandelions forming. And dandelions are a weed, so they kind of quickly take over. But their seeds are very light, and so that lets them easily be movable by wind. These seeds also can move by wind, so you can see that they kind of have like a propeller shape. I like to call these helicopter seeds. So they actually have like a heavy part of it, probably down here, and then the wind makes them kind of copter around. They like swirl around and moves the seeds far away from the parent plant. And we want to be able to disperse the seeds because the farther away the seeds move from other plants, the less competition there would be, right? So plants, this is important for plants to disperse their seeds. Some are moved by water, right? Like coconuts are buoyant. We talked about mangrove seeds being very buoyant, so water can easily move them around. Others get moved around by animals, right? So maybe an animal eats the seeds or eats the fruit, right? And they might carry it and drop the seed elsewhere if they didn't finish it, or they might eat it and then pass it through their digestive tract and then leave those seeds also kind of as waste in other areas, and they might germinate. And then some actually are able to kind of explode. There are explosive seeds. They burst open, and this will actually cause their seeds to sort of like burst out into the air and then land in other locations. This is typically caused by a lot of tension building up within the pod as the sun dries out the pod. And then ultimately, once there's enough tension built up, it will just kind of split open in sort of an explosive display. So we're going to watch this little video clip showing explosive seeds.
Violet flower seeds are crammed into a special pod. As the pod dries out, the pressure is intense. Eventually, something's got to give. Touch-me-nots are even more highly sprung. The slightest contact triggers a chain reaction. Seeds light up the sky like a fireworks display. Another dramatic display comes from this unassuming-looking plant, the squirting cucumber. It only takes a tiny vibration, and the cucumber starts a spectacular show. Seeds disappear into the distance like rockets. Spreading new plants in every direction.
Okay, in seed germination. In order for a seed to germinate, it does need proper conditions. So it's definitely going to need certain nutrients. It's going to need a certain level of moisture, so some water. If conditions are right, it will germinate. And so how does it get the nutrients it needs for germination when a seed grows underground, right? Has no leaves yet, can't photosynthesize, and it's going to need some nutrient content to be able to continue to develop and ultimately germinate. And so it's going to use that triploid tissue we talked about, that endosperm, for embryological development. In many dicot species, though, that endosperm will actually be moved into a location inside the seed that's called a cotyledon. In dicots, they have two cotyledons that they will move their endosperm into. So a mature seed is not going to have what's called endosperm anymore. It's going to have been moved into the cotyledons. And so here you can see a dicot seed, right? So think of anything that's been sort of split in half that you've seen before, like a pinto bean that you could split in half, or an edamame bean that splits in half. The reason you can split it in half is because it has two cotyledons. A monocot only has one cotyledon, so it cannot be split in half. So when you eat like a pinto bean or an edamame bean, all that kind of fleshy part that you eat that's very starchy, you're eating that endosperm, like you're eating that food reserve that a seed would be using to grow and develop. In a future class, we will dissect a seed so you can see all those parts in person. A mature dicot seed is considered to be quiescent, meaning that the embryo isn't really doing a lot of metabolic activity. It is considered to be dormant, so very, very low metabolic processes are occurring, low amounts. The seed has dehydrated most of its water content until it does not have very much water left, and so this is one of the things that allows it to remain in a dormant state for a long period of time. Remember, the embryo has these large cotyledons storing their nutrient content, and so when it is time for germination and they need to be able to have food, it will have food stored up in the cotyledons that it can use. The outer area that we said is protecting it, when the integuments harden and form the seed, that outer hard layer of the seed itself is called the testa. The testa is going to protect the seed itself. You may see other diagrams that call it a seed coat, but you need to specifically call it a testa. Here is the anatomy of a dicot seed. You are responsible only for the terms on the slide. There are other parts of a seed that if you were to Google a diagram, you may see those other parts labeled. But again, we're only covering these required parts of a dicot seed. So again, it's a developing embryo, and one of the first things to develop within this seed itself is going to be the very first root. This is called the radicle. Radicle means like first root, so it's the embryonic root. So you can see right here, this is the radicle forming, right? And so if you were doing a germination lab, for example, with seeds, you'd see the radicle emerge first. Then, of course, surrounding the entire seed is going to be the cotyledons. That's where all the food is stored. And then the shoot itself will develop, and the shoot is going to have multiple parts to it. The hypocotyl is here, and then the epicotyl is like this tippy top. You're actually only responsible for the term hypocotyl, not epicotyl. But I was afraid if I didn't show you the epicotyl, you could accidentally label the epicotyl as the hypocotyl, and that would be wrong. So really, this term is just for reference so that you know where the hypocotyl is. So the seed itself actually attaches one cotyledon to the next at an attachment point. The hypocotyl is like right below that, and then the epicotyl is above that attachment point. Epi as a root means above or on top of, and hypocotyl as a root, or hypo as a root, means below. So hypocotyl is down here, epicotyl is up here, more like where the meristematic tissue would be that's forming these first set of leaves. And then the outer area is the testa. Here's just more pictures of dicot and monocot seeds. Many seeds will stay quiescent or dormant until very specific environmental conditions occur. All seeds need water. Water is assumed, so no seed can germinate without water because seeds are so dehydrated. But some seeds actually need like crazy amounts of water to break dormancy. So when it says heavy rainfall, there's a reason heavy is bolded and underlined. That means it needs like monsoon amounts of rain. So don't say on your test, if a question about this is asked, don't just say water. That is not going to be an answer that works for this because all seeds need water. But some seeds need monsoon amounts of water, heavy rainfall. Some seeds need fire, like a red cone or a redwood cone, right? To release those seeds, it would need fire in order to do that. Some seeds need exposure to cold for a period of time before they can germinate. Like I know strawberry seeds are like that. They're really difficult to germinate. That's why you don't really see strawberry seeds at the store. You would just buy a strawberry plant for your garden because it's too hard to break the dormancy of a strawberry seed without having like a special freezer. Some seeds need exposure to sunlight. For example, some species of lettuce, they actually have to have light for germination. Most seeds don't need light. And some seeds have to be able to pass through an animal's digestive tract because inside the tract there's enzymes that can help like weaken or loosen the testa, which is going to be important for germination because eventually we're going to need water to enter that seed. Germination is going to be the process where we see the actual embryo break dormancy and develop into a baby plant, right? A seedling. And so during this time before it has germinated, it essentially is doing very little metabolic activity. Nothing is really happening inside the plant. It is quiescent, right, inside the seed itself. So it is considered to be dormant. In order for the metabolic processes to kickstart, there are certain conditions that are needed. So it will need water, but it will also require warmth and oxygen. We need oxygen for cellular respiration because the plant is going to need to be able to make ATP energy to be able to grow and develop. And we need warmth because remember, warm temperatures speed up the rate of metabolic reactions. They allow for every molecule to be moving around much faster. They have more energy. And so ultimately the reactions will happen faster. There are key steps in germination. The first one is called imbibition. In imbibition, the seed absorbs water. So this is how we kind of rehydrate the cells. Another way you could phrase this is the seed imbibes water. That means it takes in water. Imbibition has occurred. So we must rehydrate. And then some seeds, remember, have ABA or abscisic acid inside of them. One of the things that ABA does is prevent germination. So this imbibition of water will wash out all that ABA so that germination is no longer inhibited. This will also allow for gibberellins to start being produced. Remember, gibberellins is a hormone that allows for the plant to grow. That's one of the things we said that gibberellins do, is they allow for growth. They allow for mitosis. They just do it differently than auxins. So we need mitosis to stimulate so that the embryo can start growing and developing. Remember, they're going to be feeding off of that endosperm. They have all that starch stored up that needs to be broken down so that it has energy for growth. And so gibberellins is going to also help to stimulate the production of an important enzyme called amylase. Amylase is an enzyme. Remember, it ends in ase, so it's an enzyme that's going to help break down starch. And so you have amylase in your systems as well. In your saliva, amylase is like one of the first enzymes that kind of starts digestion. It's going to break down starches in your mouth into smaller sugars so that your body can easier process them. Similar in plant seeds. In the next step, this amylase will start to work. And so what amylase does is it specifically digests or hydrolyzes starch into maltose. And so maltose is a disaccharide. You can see it up here, 2 sugars, whereas starch is going to be a polysaccharide, a huge sugar, many sugars linked together. Maltose is specifically 2, and it specifically is 2 glucoses linked together. A lot of times students will say that starch is hydrolyzed by amylase into glucose, but that is not true. Amylase only hydrolyzes or breaks down starch into maltose, a disaccharide. It requires a different enzyme that can break down the maltose into glucose. You aren't required to name this enzyme, but it is called maltase. Maltase breaks down maltose into glucose, and then the glucose can be used for things. Before we talk about that, let's review some BioH vocab, right? So we have maltose here. And you can see that there's a linkage between the 2 glucoses. What is the name of this linkage that is formed in sugars that allows for us to take small sugars and build bigger sugars? Hopefully you came up with the glycosidic linkage or glycosidic bond. That's the specific bond found in sugars, in carbs. So then we are going to take our maltose and we can do different things with it. We can either break it down using enzymes into glucose, and then that glucose can be used for cellular respiration, which can then be used for growth, right, mitosis. The embryo root and the embryo shoot need to continue to grow and develop. They need to make other things from the food reserves, and so they'll need energy for all of that. So we can use the glucose for that. Or maltose can be polymerized or built together to make a different polysaccharide. We can make cellulose. Cellulose is different from starch, structurally different, and has a different function as well. Cellulose makes up the cell walls, and that allows for, strong support for the plant. In the next step of germination, there's been so much mitosis that has occurred inside the seed itself that it will rupture the testa, and then you'll see the radicle emerge. So again, rupture the testa, radicle emerges. If you were growing seeds in an IA, like in a bag, like in the lab we did, I would consider that germination. Like as soon as you see that radicle, that's the positive sign that germination has occurred. There are other ways to measure germination as well. For example, if you were planting seeds in soil, you might not be able to see germination like this, so you'd have to have other methods. So you could use probeware, right? What gas would you be looking for? What type of probeware would you have to use to show that this seed is now metabolizing? Hopefully you said carbon dioxide, because carbon dioxide is the waste product of cell resp, and that's the reaction that we'd be looking for. So you should be able to measure using probeware the carbon dioxide that's being released by the seed during germination. Or you could also look for it breaking through the soil, right? So that would definitely indicate germination has occurred. Usually when the shoot tip emerges through the soil surface, you're going to see it come through as a hook in many dicot seeds. So we'll talk about the importance of the hook. You can see like the first leaves are kind of right here. This hook shape allows for the cotyledons to protect the first set of leaves as they're coming through the surface. So what you're actually seeing come through first is the hypocotyl. Okay, so again, rupture the testa. Here's our radicle starting to form. And then here's the hypocotyl hook coming through the surface of the soil. Oops, going back. So once we have that characteristic hook shape of the hypocotyl peeking through the ground, it will eventually be stimulated by light in order to straighten the hypocotyl. So here's the hypocotyl, light hits it, and it begins to straighten. So notice as it's coming out of the ground, these are the cotyledons protecting our first set of leaves. If it was to just shoot straight out of the ground like this, without that hook shape, then the leaves may become damaged as it pushes through the soil, and we would damage our apical meristems, and that would be a problem for the overall growth of the plant as well. So once we have our first set of leaves like this, there's no more food left in our endosperm and we don't need it. We've used it all up. And so now those leaves are responsible for photosynthesizing. And that way the plant itself can have everything it needs to go through primary growth and secondary growth and so on, like we've talked about in previous sections. Before I move on, I want you to think about what would happen to the plant if you planted your seed too deep. Right, so we have our seed, it needs to be planted at a certain depth. There's always instructions on seed packages. Why is that important that you actually follow that through? Hopefully you came up with if you plant it too deep, then it's going to run out of endosperm, right? So that hypocotyl hook is never going to break through the surface. And then you won't actually have your seed be able to grow into a plant. It gets stopped under the ground. And it won't be able to get leaves for photosynthesis, so it will just die. I'm going to show you a couple of clips that will show you the hypocotyl. Actually, the first one, it doesn't show the hypocotyl coming out, but it will show germination, and then the second one will show the hypocotyl. So you can see the radicle emerging, so germination has happened. And you'll see the hook go towards the surface, but I don't think we'll see it break through. So in this one, it will go through a couple dehydration rounds, so they forget to water it. But here we go. We have germination happened. You can see the radicle going down. A little bit of a pause because they forget to water. Now they're watering again. And you'll see the hypocotyl come up. Light stimulates it to straighten. Forget to water it. And then it comes back. Okay, last thing. So I'm going to play the next slide, and I want you to just think, what is the very first thing you see on the next slide? The very first thing. Was it the deer? A lot of students say the deer. So there's this term called plant blindness, which kind of means that we're very aware of the animals around us, right? So we think animals are cute, we think they're fun, sometimes we think they're gross. Either way, we're aware of them. But many people tend to be less aware of plants. And so if that's you, you may have plant blindness. So hopefully, I'm hoping that, after this unit, and other units we've done this year, you really realize how important plants are. I'm sure that you knew plants provided important oxygen for us that we breathe, but now I think you know that plants are much more important than that, right? We would not have the nutrients that we need to enter the ecosystem if it wasn't for the plants, right? So we need plants for nitrogen, we need plants for phosphorus. They get these important nutrients into the ecosystem and into the food chain, and ultimately we depend on that for us to get our nutrients too, right? We also depend on plants to be a major part of the carbon cycle. They're just really important organisms. So this is my last pitch to hopefully make sure you're aware of the importance of plants and that you try not to have plant blindness. And this is the end of our plants unit.